Proteins. Proteins are heteropolymers and they contain a string of many amino acids. Now when we say this, it means these amino acids are joined together and a peptide bond which is formed between the carboxyl group of one amino acid and the amino group of a successive amino acid together. This bond formed between the carboxyl group as well as the amino group of two successive amino acids is known as peptide bond. And various types of proteins are formed from 20 amino acids. And these are based on specifically the number of amino acids. And number 2, the sequence of amino acids in which these are arranged. We can say as an example, collagen. It is the most abundant animal protein that means a protein in the animal world, and ribulose 1,5 biphosphate carboxylase. Carboxylase oxygenase, we call it ribisco. It is the most abundantly known protein in the biosphere. Okay, this collagen is in the animal world, but this is in the entire biosphere. Most abundant protein. There are some proteins and their functions are associated along with. Supposingly we say collagen. Collagen, its function is it acts as an intercellular and extracellular ground substance. Then we say trypsin. Trypsin is an enzyme to digest protein. Then insulin. It acts as an hormone to regulate the blood glucose level. Then gamma, an antibody. What an antibody does? It fights against infection. It's the gamma globulin. Then there are certain receptors and these receptors, basically these are the proteins that receive the sensory impulse. Sensory impulse like smell, like taste, etc. Then is GLUT4. It regulates transport of glucose in cells, into the cells. Now coming to the structure of proteins. When we come to the structure of proteins, We say its structure is basically the three-dimensional structure of macromolecules, and on the basis it can be primary. Primary is basically the basic structure. Basic structure means the sequence of amino acids. Sequence of amino acids in a polypeptide. Okay, and this sequence of amino acid, it describes or determines the place where the polypeptides are used and from where these have to bend and where the different lengths will be attached to each other. So here, let's start with the basic structure. Here, this is the first amino acid. This is the second amino acid. These are linked together with this peptide linkage. C double bond O N H. This is the peptide linkage. Now, the secondary structure. Secondary structure is the development of steric relationships. Of amino acids. Basically, these are of two types. These are alpha helix and beta pleated. Alpha helix, it's in the form of basically coils or this kind of spring-like structure, and sheets sort of this manner. So here the groups, that is the residues, all the main chains having the Cu and NH group of hydrogen bonds, these are coiled, and secondary, this kind of alpha helical one, it is found in hair, that is the keratin of the hair, myosin, tropomyosin, these are the muscles if you remember. Then epidermis also, then in the fibrin of the blood clot. Here the polypeptides, they coil around each other to form these cable-like networks. In beta pleated structure, the chain gets interconnected by hydrogen bonds and a sheet is produced instead of a fiber or a rod. Therefore, this structure is called pleated sheet structure, and it is normally found in the fibroin of silk, and this is having a different structural similarity compared to this one. So here in this particular one, we observe the bonding as NH2CH, C double bond O, alright, and then same NH of another one, another group. Here when these are bending and these are moving like this, then the back side, the front side, the back side, the front side, the back side, front side. So in this back side, while moving like this, when the C double bond O NH group of the third one comes and joins, C double bond O and NH, This NH and this CO, they form a bond together. And how? The structure is tightly held in this alpha helix is only because of this